Hi, my name is Mihika, and I'm going to be talking about clustering in bioinformatics. So let us understand what clustering is. In clustering algorithms, which is an unsupervised learning algorithm, what does unsupervised learning algorithm mean? It means that we don't have an output variable to train our data against. And so, with clustering algorithms, what we hope to do is to group data or group genes with similar expression patterns into clusters so that these clusters can correspond to groups of functionally related genes. Let us take an example of the same. Now, as biologists, we can take DNA arrays, which allows us to analyze the expression levels, which means that the amount of mRNA that is produced in a cell of many genes under many time points and conditions to reveal which genes are switched on at a particular time period. Now, through this study, we come out with this intensity matrix over here. Now, this intensity matrix in this example has n rows and m columns. The n rows over here correspond to the genes that are present in the cell, and the m columns correspond to the particular time period or the condition where the genes are switched on and off. This expression matrix thus represents the intensity of hybridization signals produced by this DNA array. Now, we can see very clearly that there are certain genes that form pairs as they have similar expression patterns. And so, we can somehow conclude that these genes are maybe similar. Maybe they have similar functions or are involved in the same biological process. And so, accordingly, if a gene is, or if there's an expression pattern of a newly sequenced gene that is similar to this, then we can kind of suspect that these genes perform similar or related functions. And so we can cluster these genes into one group to kind of conclude these assumptions that we have. Maybe these genes are similar. And so to conclude that, we can cluster them together. Like we can see gene 3 and gene 5 in this particular matrix have very similar expression patterns. And so they can be grouped into the same cluster. To do this clustering, we map our intensity matrix into an n into n distance matrix. And this distance matrix helps us understand how similar the expression patterns of these genes are. And these are when they're plotted onto a graph of this sort, we can apply a certain distance metric like Euclidean distance or Manhattan distance to understand how close these points are to each other. And the closer they are, they can form a cluster, while the further they are, they can form two different clusters. And this is based on two main conditions. These conditions are homogeneity and separation. So as the word suggests, homogeneity means that the data within one cluster should be highly similar, while separation means that the data between two different clusters has to be completely different. Let us understand this with the help of a graph. This graph clearly shows good homogeneity and separation. Like you can see 2 points over here, they are apart, they have a lot of distance between each other. So we can assume that these two data points or these two genes are completely different in characteristics, and so are part of two different clusters and show good separation. And since these 2 points that are very close to each other, we can assume that they have the same characteristics and so are homogeneous and thus are part of the same cluster. But in the second graph, as you can see, it shows very clear bad homogeneity and separation, because these 2 points over here clearly have a lot of distance between each other and still are part of the same cluster, which means there is no good separation between these 2 points. And these 2 points, for example, are very similar. They don't have a lot of distance between each other. So we can assume that they are similar genes or similar data points and still are a part of two different clusters. And thus, for our clustering algorithm to be efficient and perform in the best way possible, we need good homogeneity and separation, especially when we have large amounts of data or gene expression microarray data that we use in bioinformatics, We need good homogeneity and separation in our clustering algorithms. So these are the references that I referred to for this presentation. And if there are any doubts, you can always contact me on my email ID. Thank you so much.